हे एवरीवन, वेलकम टू बायोलॉजी पाठशाला एंड एनिमेशन चैनल। तो आज मैं लैब में आया हूँ। तो आज मैं आप लोगों को एक इंस्ट्रूमेंट का यूज़ सिखाऊंगा कि उस इंस्ट्रूमेंट को कैसे यूज़ करते हैं। तो चलिए वीडियो में देखते हैं मैं कौन सा इंस्ट्रूमेंट सिखाता हूँ आप लोगों को। So today we are going to learn about this instrument which is known as colorimeter. Colorimeter is an instrument that is generally used to determine the absorbance or optical density of a given solution by at a particular wavelength of light. This instrument is used to determine the concentration of a known solute by the Beer-Lambert law. According to Beer-Lambert law, the optical density or the OD will increase with the concentration of the sample. So here this is a colorimeter and it need to be heat up for 30 minutes. So I am plugging in the charger and I am making the machine switched on and it will heat up for 30 minutes. These are known as cuvettes. Generally, we will use these cuvettes to take the sample and put it into the colorimeter. We need to handle these cuvettes very carefully and we should make sure that we do not touch the lower portion of the cuvette because light will pass from here. Now this is our sample. One sample is a blank. This one is the blank sample which contained ethanol, and the other one which is green in color, that is containing the chlorophyll. That means that is the leaf is crushed with the ethanol and is centrifuged to get that supernatant. We will determine the OD of the supernatant. Now here it is the colorimeter and we have various wavelength of light that is written in the above of the colorimeter. So there is nanometer also given, that is 400 from 420, that is violet, and we need to adjust the particular wavelength we are looking for to determine the OD by moving this. And here you can see that I am moving that and I am giving the 620 nanometer, which is for greenish and yellowish, because I will determine the OD of chlorophyll. So here I am working on the colorimeter, and first of all I am taking the cuvettes out of its box. So after taking the cuvette, I am filling the cuvette with the blank, that is ethanol. Here you can see that is the blank solution, that is ethanol. So after taking the ethanolic solution in the blank cuvette, I am putting that in the colorimeter and I will set 0 for the blank, so the absorbance of the blank will be 0. So here the blank absorbance is done on 0, and now I will determine the absorbance of the sample, that is the crushed chlorophyll. So I will wash the cuvette and I will clean it with a tissue paper properly. Now I will take the sample, that is the crushed chlorophyll. So after taking the sample, I will put it on the colorimeter and I will see what it gives, the optical density or the absorbance of it. So the colorimeter has given 0.26 optical density for the chlorophyll against the blank of ethanol. If the video was helpful, then please share and subscribe.